In this week's lab, we're going to do 2 different experiments, and both of them are going to be based on the scientific method. You've probably heard about the scientific method before, and you've probably run little experiments in, as for science fairs and that kind of thing. So, and you should know that scientific method starts with an observation. Based on that, you formulate a hypothesis, which is an educated guess. It's important for a hypothesis that it's falsifiable, that you can figure out when it's not working. Then you do an experiment, you collect data, you analyze the data. If the evidence supports your hypothesis, you communicate to others that it supported your hypothesis. And then this hypothesis gets you try over and over again, and you have to make sure that other scientists can repeat this. You're seeing this in action right now with COVID-19, where we make observations and then we draw conclusions and then we revise these conclusions. And to a layperson, oftentimes this looks pretty weird because, like with COVID-19, we at first looked like it was a respiratory virus, but now it's looking like there's evidence that's coming out that it's maybe not a respiratory virus. And the most important thing a scientist can do is to change his or her mind. So you always have to be willing to, when you have better data, you change your mind. And that's very important in science. Now, if your evidence refutes your hypothesis, then you revise your hypothesis and you come up with a different experiment and a different idea, a different explanation. What we're going to do, we have a hypothesis. Our hypothesis is to say that it is possible to detect important molecules based on their reaction with simple indicator reagents. I talked in the lecture about the indicator reagents and how they work. I also talked about the macromolecules in the lecture part. And we're going to explore these macromolecules now further. Now, what we're going to be focusing on this time around is not so much about the scientific method, but about the experimental design. How do you set up an experiment? Because as it turns out, the devil in science is always in the detail. And so what I want to specifically talk about when it comes to the experimental design, you have to pay attention to the groups that you're doing this experiment on, and the variables, and how you're controlling them, and that. So when you're looking at the groups, there are 2 sets of groups you've got to be thinking about. One is the control group. That tells you what it looks like when it works, when it's a positive result. Like in our case, is we're looking for a color change. And in the positive case, you see the color change. In the negative case, you don't see the color change. So if you're going to test for protein, you always run a control with your experiment where you always have something that has protein in it and something that doesn't have protein in it. So you have something you can compare it to. And then your experimental is going to look either like the positive, which means that it would have protein in it, or it'll look like the negative. Then the other thing you got to pay attention to is your variables. And ideally, in an experiment, you're holding most variables constant. So you're going to try to make sure that they're really only one variable that you manipulate. That is the independent variable. That's the variable that you change, the variable you manipulate. So, and the dependent variable is the variable that you measure. You can't do science without measuring something. So measurement could be a distance, a weight, whatever, something that you can, something that changes, something you can measure. In our case, we're measuring a color change. And all the other variables we're going to try to hold constant. This is why science with humans is really difficult, because trying to hold everything constant, because humans do what humans do and they're different. It's hard to keep lots of things constant, which is why when you're doing human research, you need lots of numbers, so that the constant is within a range of what you can predict. Now, to remind you again of the tests we were looking at, we have 4 different tests that we're going to be looking at. There's the test for Benedict's, which is the Benedict's reagent, which tests for reducing sugars. And if you remember there was copper in that solution, and when copper is reduced, it turns orange. And if it's a lot reduced, it turns orange. If it's reduced just a little bit, it can turn green. Then you had your iodine solution that tested for starch. Did I mention the Benedict solution tests for reducing sugars? The iodine solution tests for starch, which is a carbohydrate. It's a polysaccharide. And if there's starch present, if you add iodine to the solution, it turns black. So a positive result is black. And then you have burette reagent, which tests for protein. It's a light blue color when it starts out, and if there's protein present in the solution, it turns purple. And then last but not least, we have Sudan 4, which is our test for lipids. And if lipid, it's a lipid soluble dye. So if your reaction is positive, it will be the dye dissolves in your test solution. And if it's negative, you'll end up with 2 layers. So if we're going to try to combine those 2 things together, let's think about for example, in the burette test, which is the test for protein, and the positive. So what's the positive supposed to look like? What's the negative supposed to look like? And how do we set up this experiment? What are the variables that are independent? What are the variables that are dependent? And if you set this up, the positive is a purple color. The negative is just clear or light blue, which is what the reaction was to start with. And then when we set up the control reagents in the lab, we try a couple of different things. We try with water. We try to see what it looks like with protein. We try to see what it looks like with starch. And we try to see what it looks like with lipid. In each of these cases, we're adding 12 drops to a solution. And then we use the same test solution on these. So the water, protein, starch, and lipid, that's the variable you change. So that's it's independent of the experiment, because it's your input. It's how you're setting the experiment up. It's not what you're measuring. Then you're adding the same amount of burette reagent to everything, 12 drops for everybody, so that you have a constant volume. You're doing this in the same kind of tubes. You're doing this together in the same environment, so that you can compare all of these to each other. And that what you're measuring is the color change. And your expectation would be, because burette tests for protein, your expectation would be that there would not be a color change in the water, there would not be a color change in the starch, there would not be a color change in the lipid, but there would be a color change in the protein. So your water, your starch, and your lipid would all be negative controls, whereas your protein is your positive control. And then you would be comparing your unknowns to those results. So to remind you again what these tests test for and what they look like, the Benedict's test tests for reducing sugars. Your positive results are going to be green or purple, green or orange, with green being less sugar than the orange is. Your negative result is going to be a light blue color, which is how it starts out. The iodine test, the negative control is going to look kind of yellowish because that's the color of iodine. And if starch is present, it turns black. The burette test tests for protein. The negative is a slightly bluish color. The positive is purple. And then the Sudan 4, as I mentioned before, it becomes red throughout if it's positive, and if it's negative, you end up with 2 layers because lipids are known for the fact that they are hydrophobic, so they don't mix with water well. When we do this in the lab, we spend the most amount of time with this setup, actually. I want you to look at this in case we have the ability to actually go in the lab so that you can set this up. And the way this is set up, and I want you to think about what on here are the positive controls and what on here are the negative controls. So we have if we do this in the lab, you end up with a number of different tubes. Everything that gets Benedict's would be labeled with A. Everything that gets iodine would be labeled with B. Everything labeled with burette would be C, and everything that gets the Sudan 4 would be labeled with D, and then you would add water to everything that has a 1. You would add glucose, which is a reducing sugar, to everything that has a 2. So A2 would have Benedict's and reducing sugar. Everything that has a 3 has starch in it. And the reason why we're setting this up this way is because some of these reagents that we're using are actually hazardous. The Benedict's and the burette are hazardous solutions. So when we toss these, we need to make sure that the right reagents go into the right kind of hazardous waste. And so by setting it up this way, everything that's labeled with an A goes in the Benedict's trash. Everything that's labeled with a B goes in the iodine trash. Everything that's labeled with a C goes in the burette test, in the burette trash. And everything that's labeled with a D goes into that trash. So that way we're keeping track of what reagent actually was in everything. So I'm going to give you a second now where you can pause the video just for you to think about for a second what is the positive and the negative control in each of these cases. I'm going to give you a second. So you want to stop the video right now if you want to think about it a little bit longer, because I'm getting ready to switch to the next picture. So this shows you where you're expecting a positive change. So the water is going to be a negative control for all of these different tests. Benedict's is expected to change color with a reducing sugar, which is what glucose is. Starch is a carbohydrate, but it's not a reducing sugar. The iodine test tests for starch, so you're expecting a black color with the starch. The burette test tests for protein, so you're expecting purple with the protein. So C4 is going to be your positive control on that one. All the others are negative controls. And the lipid is the one where you're expecting a positive result on the Sudan 4 test. And that brings us to the unknowns. So in your lab handout, and I'm going to switch real quick to your lab handout. Actually, we'll go through this first. We'll do this first real quick, and then I'll go to the lab handout. So in the lab handout, you'll have a picture like the picture down here, which is your unknown number 1. The first tube shows you the appearance of the unknown without any additions. The second test shows you the appearance of the unknown with the Benedict's test. The third one shows you the appearance of the unknown with the iodine test, and then the Viret test, and then the Sudan 4 test. On the top here, I gave you a reminder again what the negative and the positive look like. And so what you have to indicate in these boxes is which one is a positive and which one is a negative test. So, oh, and I know you cannot see my pointer. I need to turn the pointer on. So what you need to then do is, so the appearance of the unknown is clear. Then here on the Benedict's test, is this a positive or is this a negative? So I'm going to put this into the pen, and hopefully I can write this. So the Benedict's test, you see the Benedict's test up here, right? This is your Benedict's test, and this is your negative, and this is your positive. So the Benedict's test would be a positive test. Now on the iodine test, this looks, because the black would be the positive, it looks like a negative test. The Viret test looks like it's a negative, because the negative, the positive would be purple. And the Sudan 4, you have 2 layers, so that would be a negative test. So the only thing that's in here is a reducing sugar, and you can think about what this possibly may be. It's a clear, it doesn't contain anything but the reducing sugar. So, well, I'll give you a list of possible things it could be later on, and then hopefully you can mix and match and see what that is. When we get to the next one, the next sample that I wanted to go through, you do want to look at the unknown, what it looks like. In the lab, we have the advantage of actually being able to smell the unknown. And some of you who know your sodas very well, you can see well, you can smell and you can see the Mountain Dew all the way across the room. So this unknown is white. So that should give you a little bit of an idea what it may possibly be. And then we're going to look at the Benedict's test. Now the Benedict's test is listed up here as orange, but remember the Benedict's test was a reducing test. It's for reducing sugars. So there's copper in here, and copper, as we talked about in the lecture, changes color. Remember I told you about the copper penny that turns green first? And so what you have here is you have a reducing sugar that went green, but didn't go all the way to the red color. So this green is also a positive, but it's a positive. It's less positive than the red would be. So there is a little bit of sugar here. Plus, the iodine test, there's not starch in here, but it looks like there's protein in here. And this one does not really show a complete layer. This is throughout. It got mixed, and then there's white shit that came out, but this is mixed throughout. So what do you think this could possibly be? Something that looks white, that has a sugar of some sort in it, a reducing sugar of some sort. And remember that all monosaccharides and some disaccharides are reducing sugars. It looks like it has. So this looks like it's a pretty complete kind of, kind of nutrient. It's got a lot of things in it. And so now the next thing we're going to do is I'm going to stop this real quick, and I'm going to share with you the handout. So the handout is the thing you're going to submit. Share this now. Come on. Oh, come on. Share it. So the handout, the lab handout or the lab activity sheet contains the information where you're going to fill out different things. Now, if you can't see this, hang on, let me enlarge it a little bit so I can see it better. So the first, so here are the things I want you to fill out. So the first thing you see here is the test for reducing sugar. Listed here, the test for reducing sugar. And these are your controls. So you did the test tube with 12 materials to be tested, which was your substrate. So your substrates are distilled water, 10% glucose, 1% starch, 1% ovalbumin, and corn oil. I've listed on your setup pages that the starch obviously is starch, but the ovalbumin, that's your protein, and the corn oil, that's your lipid. So with the Benedicts, your positive reaction is only with the glucose, so that is your positive control. So which one of the tubes is the negative control? That would be all the others. So you just list the tubes that are the negative control here. And then what the color of the negative control is the one that didn't change, right? Which tube has evidence of a reducing sugar? You just put that number in there, and what it looks like, again, just write that in there. Which tubes don't react with the Benedict's reagents would be the ones that don't have reducing sugars. And what is the evidence that this test is specific for reducing sugar and not all carbohydrates? Well, one of these things is a carbohydrate also, right? That is not a reducing sugar. So it's specific for reducing sugars for that reason. The next test, actually, let's look at this one first. This one is another, is the same test, it's the Benedict's test again, but it's looking at the more at the specificity and the amount of glucose that's in there. So we talked about the fact that sucrose was not a reducing sugar. It's a sugar, but it's not a reducing sugar. And you see here, you don't have a color change with sucrose. With 1% glucose and 10% glucose, this is semi-quantitative. So you can tell that with 1% glucose, you only go to green from the blue. You don't go all the way to the orange. So, and you explain here why this is the case. The next test then is again, you're going to answer these questions and then you're going to submit this. And so you're going to make your way all the way through, first through all the controls, and then we get to the unknowns. We just walked through the first unknown, complete with answers, and the second one. And what I want you to do is I want you to run through the rest of them. So there's 10 unknowns altogether. And make sure that you look at what the unknown looks like and then what the results are. And you're going to make your way all the way through all of the unknowns, and then you get to a page where, based on the composition of the unknown results, you should get you can make a guess of what those foods may possibly be. One of the things I'm going to do is I'm going to give you a quiz where you can match. So I'm going to give you 10 unknowns, and they will match. If you do the quiz, you can do with matching, figure out which unknowns are which, because it's a lot harder to figure this out if you're actually able to smell these things. When we do this in the lab, we actually, one of the unknowns is light beer, and it's a lot of fun to smell the light beer. And then last but not least, we're going to do, you have a reaction on the, for the testing of GMO, and we're going to look at what has a GMO in it. So there's an experiment that Larry does where he shows you how to do this, and then you use this. You extract these samples, and then you look at this antibody test, where you can see whether something has a GMO present or not. And I'm going over that test separately in a PowerPoint, just to give you a little bit of an idea how these kinds of tests work. Because they're actually they're quite interesting as tests goes, and you, especially those of you who are female, may, when you're looking at the test, be a little bit reminded of what a pregnancy test looks like, and because that's also an antibody test. But yeah, so that's going to be a separate PowerPoint compared to this PowerPoint. So, and I'm hoping that that's going to tell you a little bit about what you're supposed to be doing in class today. And you're submitting this this lab sheet basically. And that was all I wanted to go over for the lab.